Okay, so this is the second part where we're talking about DNA and genes. Specifically, we're going to talk about a few more genetic conditions, sex-linked traits. And then we're also going to talk about how a gene goes from DNA, it's transcribed into RNA, and then translated into a protein. Then we're going to start with another historical figure. And Thomas Hunt Morgan, who was born in 1910, he worked with the fruit fly Drosophila melanogaster, which normally has red eyes, but he found that some of these males had white eyes. Okay, so to figure out whether it was a dominant recessive trait, he mated white-eyed and red-eyed individuals and found that all of the F1 progeny had red eyes. So we know it's a dominant trait. And then he took the F1 generation and he crossed these F1 females, which had red eyes, and F1 males, which had white eyes. Well, actually, they all had red eyes at this point. All of the F1 had red eyes. What he found in the F2 generation was that he had both red and white eyes, but all of the white-eyed flies were male. When he did test cross of F1 females, he did find some females that had white eyes. But so that was a possibility, but he didn't understand why most of the F2s were, well, all of the F2 generation that had white eyes were males. Until he thought about the sex chromosomes, and he concluded that, well, the gene for eye color must be on the X chromosome. The reason why this makes sense is because the Y chromosome is a little bit smaller than the X chromosome. Well, it's a lot of bit smaller than the X chromosome. In humans as well as in flies, sex is determined by the presence of an X or Y chromosome. If you have two X chromosomes, you're female. If you have an X and a Y, you are male. Humans have 22 autosomal, meaning non-sex chromosomes, and then a pair of sex chromosomes. Humans and flies have the same XY system. Birds have an opposite system where the male has both, is the homozygous, sex chromosome, and the female has a shorter chromosome, heterozygous. Grasshoppers, if you have two X's, you're female, one X, you're male, and then honeybees do a haploid diploid. So there are a few traits that are sex-linked, which are on the X chromosome, and they affect males to a greater degree than females. A few of these are red-green colorblindness. So this test here is to see if you're red-green colorblind. Males, because it's on the X chromosome, are more often red-green colorblind than females. So if you can't see that there is a sailboat on this, and you can't see the number 71 on this, then, or 74 actually, then you are probably red-green colorblind. And there is a form of hemophilia that also is an X-linked recessive. And so most people who have hemophilia are males because it's on the X chromosome. And then most females are carriers if they can exhibit normal function. Hemophilia is a disease where if you start bleeding, you can't stop bleeding. You don't have the clotting factors in your blood. Females can be heterozygous for this and are fine, but if males have an affected allele because they only have one X chromosome, then they are more likely to have it. And you can see this, we have a pedigree chart here, and you see affected individuals which have hemophilia, and this was in the royal family, continued to be propagated in the royal family because of Queen Victoria. She was the first one who had it, and then passed it on to much of her progeny. So, to compensate for only having one X allele, females will turn off one of their X chromosomes, just like males. And this is done at random, so it's not the same X chromosome that's turned off in every one of their cells, and this is what leads to some things like in cats that have different colors. The different colors are because of these inactivated X chromosomes, which are called Barr bodies. And so females, all females, are basically genetic mosaics, meaning they have different genes being turned on different parts of their body, depending on which Barr body or which X chromosome was made into a Barr body. So there are a few exceptions to the idea that all our characteristics are determined by the chromosomes in the nucleus, one of them being mitochondria and chloroplasts have their own DNA. So they don't follow the same rules that our chromosomes in our nucleus do. Genes from the mitochondria and chloroplasts are passed from generation to generation through the females, which is called maternal inheritance. So once we figured out that there's different types of genes, geneticists realized that they could learn how far genes were or how close they were on a chromosome by the amount of crossing over. And if crossing over occurs, then the parental alleles are recombined, so producing a recombinant gamete. Okay. And Alfred Sturtevant, who was a student of T.H. Morgan, observed that the recombinant progeny reflected the location of the genes relative to each other. So if two genes are on the same chromosome, so let's say blue eyes or eye color and hair color were on the same chromosome, and they were close to each other, then they would be associated with each other very often. But if they're further apart, then they wouldn't be as associated with each other because of crossing over events. So Sturtevant used this theory to basically come up with, well, if you look at the crossing over events, you could determine how close those genes are to each other. And he came up with this recombination frequency, which equals the recombinant progeny divided by the total progeny. A 1% recombination was then referred to as one map unit. And we'll do some, or another unit would be called the centimorgan. And we'll do some of these problems, and it'll make more sense when we go over the problems in class. So another way to look at how genes are relative to each other is by doing a 3-point test cross. So this looks at 3 different genes, or 3 different locations, to construct a map of where these genes are located relative to each other. So by doing a 3-point test cross, I can tell you if A is closer to C than it is to B. And again, we'll do some problems like this in class. People have used these test crosses to look at genes on the same chromosome that are for different diseases. But there weren't really enough markers to do a very dense study of all these genes until the 1980s. And once we developed anonymous markers, the techniques for determining the lengths between genes or between locations on the genes became a lot easier. Another advent of genetics that made things a lot easier for genetic mapping was the advent of SNPs, or single nucleotide polymorphisms, which is basically a difference in one nucleotide. And this affects a single base, but is used to increase the ability for mapping genes. This is used in forensic analysis. You look at the base pairs on different genes, different DNA from different individuals, and if they match up, then it's most likely going to be the same individual. also used for paternity testing. So, sickle cell anemia was one of these diseases that was the first one to show the result of a mutation in a protein. And this was caused by a defect in hemoglobin, which we know is used to carry oxygen in the blood. And when you have sickle cell anemia, the shape is like this one here. And because the shape is different, it's not able to carry as much blood, and it also clogs arteries, and so it causes a lot of problems. If you're homozygous for sickle cell anemia, then you'll have very impaired health, and usually they have a reduced lifespan. But heterozygotes appear normal, probably because they have some normal blood and some sickle cell blood. One benefit of this, though, is that the parasite that causes malaria isn't able to infect the sickle cell blood. So heterozygotes do not contract malaria, and so you see a lot of a greater prevalence of these of sickle cell anemia in areas where malaria is prevalent, which is sub-Saharan Africa and southeastern Asia. Some of these areas here have a lot of high incidence of sickle cell anemia because it helps against malaria, which is a very devastating disease. All right, we briefly talked about this before, but non-disjunction is where during meiosis the chromosomes do not fully separate, do not go to evenly to both sides of the cell. And aneuploidy is then gaining or losing a chromosome. If you have monosomy, that means you lost a chromosome. If you have trisomy, that means you gained a chromosome. This here is trisomy 21, which is Down syndrome. And in shorter autosomes, like 21, it seems to you're able to have 3 copies and still survive. But usually, if you do on one of these larger ones, like 1, 2, or 3, it's most likely going to be fatal, and the zygote would not produce a fetus that would, the fetus would probably abort, and you wouldn't have a full-term baby. Excuse me. Now, non-disjunction in the X or Y or sex chromosomes can cause some severe developmental abnormalities, but also some of them can be very viable into a later age of life. So, for example, you have XXX females. XXYs is called Klinefelter syndrome, and these are males. XO is called Turner syndrome, where they only have 1 X chromosome. And OY, if you don't have an X chromosome, there are too many genes on the X chromosome necessary for life. So if you are OY, then you will not survive to become a full-term fetus. If you have 2 Ys and 1 X, that's called Jacob syndrome, and there are a lot of complications with that as well. Alright, so genomic imprinting is where the phenotype of the expression of a gene is affected by whether the mother or the father contributed the allele to the offspring. So, for example, if you have a partial deletion of chromosome 15, if it's from the father, you'll have Prader-Willi syndrome, and that's what this is to the left. It has some developmental issues in the skull and in the fingers, and usually results in an increased appetite for food. But the same chromosome, if it comes from the mother, is called Engelmann syndrome, and it produces a host of different abnormalities. Epigenetics is changes in the DNA and histones that can be inherited. So this is not inheriting a gene from your mother or your father, but inheriting maybe the gene that is turned on or off. And this is the idea that not everything's in the genes, but the proteins and the processes surrounding the genes can also affect the expression of DNA. DNA methylation is a process where different molecules are put on the DNA and they will turn off or on genes. And the DNA can also be changed based on these proteins that surround the chromosomes. So we see evidence of this if you look at chromosomes of identical twins when they are 3 years old. It will show almost the same pattern of usage of their chromosomes. But when you look at when they're 50, there are a lot more differences in their chromosomes. And that's just because environmental factors also affect the expression of genes. And these can also be passed from mother to father. There's a lot of research being done on that. So you can also detect genes while a fetus is still growing within the mother's uterus. But there are a couple other techniques you can use as well. One is a pedigree analysis, so you can look at the rate of infection among your parents, cousins, uncles, grandparents, so on and so forth, back forth, to see if you have a propensity for a certain disease. A couple of kind of invasive procedures, an amniocentesis is done on fetal cells collected from the amniotic fluid. So they pull some amniotic fluid out, they look at the cells. Some of those cells belong to the baby or the growing fetus, and they do a DNA analysis on them. Chorionic villi sampling is a little bit more invasive, and that's where they collect cells from the placenta and then do a DNA analysis on those. So once they have that DNA, then they can screen for certain genetic diseases or whatever they want, really. Another procedure is called pre-implantation genetic diagnosis, and this is kind of a cool technique. Basically, in vitro fertilization, they take embryos or they take eggs and they take sperm and they fertilize them outside the womb, and then they do they take one cell out of the developing embryo and they do a genetic analysis of it, and then they can tell the parents which of these embryos they would like to implant back into the uterus. It can also be used for gender, or basically any trait that you want to screen for, you can look for. But there are some ethical questions surrounding whether you should be able to pick and choose what traits you want your babies to have. And we'll watch this video in class of how it takes place. Alright, so then the next part of this is going to be the process of transcription and translation. So the central dogma is this process, which we've kind of hinted at. We talked about organelles, we talked about the ribosomes, but it's the process of taking DNA, transcribing it into RNA, and then translating it into protein. So transcription is the DNA to RNA process, and translation is the RNA to protein. And this was first described by Francis Crick, who also helped with the discovering the shape and structure of DNA. So transcription is DNA-directed synthesis of RNA. So it's done on the DNA, and you have enzymes that make copies or sections of messenger in our RNA. As we discussed before, uracil in DNA is replaced by thymine in RNA, and then mRNA is used to direct the synthesis of polypeptides. Translation then is that synthesis of polypeptides or proteins, and this takes place at the ribosome and requires several kinds of RNA. We have messenger RNA, which is composed of base pairs that pair with DNA. We have ribosomal RNA, which is part of a ribosome. We have transfer RNA, which is involved in the transcription process. And then we have a few other small nuclear RNA, signal recognition particle RNA, and microRNA, which we aren't going to go over. So the genetic code, Francis Crick and Sidney Brenner determined how this code then led to the amino acids. A codon then is a block of 3 DNA nucleotides corresponding to an amino acid. So if you see here, you have AUG, these are adenine, uracil, and guanine. This codes for the amino acid methionine. And the way that they figure this out is they would introduce single nucleotide insertions or deletions and then look and see how that changed the amino acids. And this indicates the importance of a reading frame, that it was continuous, there was no spaces between these. So if you deleted a base, it messed everything up. So there are specific codons that do certain things. Marshall and Nirenberg discovered the stop codons, and there are 3 of those, UUA, UGA, and UAG. There's a start codon, AUG, which signifies the start of translation, and then the rest of the combinations code for amino acids. The code is degenerate, which means that some amino acids have more than one codon. So for example, UUA, UGA, and UAG, they're all slightly different, but they all code for the stop codon. And this code is practically universal. So there's really very little difference between the codons used in plants, as there are for animals, or there for bacteria. They all use the same codons, and they all code for almost the exact same amino acids. And here they are. So these are all the amino acids, and these are their different codes. So if I were to give you, let's say, GCU, what amino acid does that code for? You could go here, the first letter would be G, the second letter would be C, and then the third letter would be U, and you would know that's for alanine. So we will do some activities with that in class as well. Alright, transcription is a little different in prokaryotes and eukaryotes. In prokaryotes, you have one RNA polymerase, and RNA polymerase is an enzyme. Anything that ends in ACE is an enzyme. RNA polymerase then adds polymers of RNA and is involved in the initiation of messenger RNA synthesis. It doesn't require a primer, but does require a promoter, a start site, and a termination site. And that is called then the transcription unit. So the promoter is a site of specific DNA sequence that is recognized that the RNA polymerase binds to, and it's usually upstream of the start site. And it's not transcribed, so it doesn't turn into a messenger RNA. It just codes is a place for where the RNA polymerase will bind to. And it's asymmetrical, which meaning it is not even. And that way the RNA polymerase knows which end to bind to. So you have an RNA polymerase here. Upstream of that you have the promoter, the start code, and then where it starts binding for the RNA polymerase is the promoter. Alright, so then after the RNA polymerase is bound, you have elongation, and this grows in the 5' to 3' direction. So again, this is talking about the direction of the amino acids. Sorry, not amino acids, of the nucleotides. So nucleotides are added in the 5' to 3 on the 3' end. You add a new one, and this creates a transcription bubble where the DNA is unwound and the transcription occurs. Once the bubble passes, then it rewinds and rebinds back together, and the messenger RNA is then continued to kind of lag behind that bubble. Termination then is the final step, and that is marked by a sequence that signals stop. Okay, the polymerase gets to the sequence, okay, we're done transcribing. And the phosphodiester bonds between the nucleotides are no longer made, and an RNA-DNA hybrid with the transcription bubble dissociates. So the RNA starts falling off the DNA, and the RNA polymerase lets the DNA go, and the DNA coils back up, and then the RNA produces this hairpin where you have binding of base codes of base pairs across the RNA, and that's what causes the polymerase to pause and to let it go. Okay, and then prokaryotic translation is also a little bit different, and it is tied to transcription, so you don't have to wait for transcription to complete before translation can begin. But it requires an operon, which is a group of functionally related genes and enzymes that are regulated together. Okay, there are 3 different types of RNA polymerases in eukaryotic transcription. They're a little more complicated. RNA polymerase 1 transcribes ribosomal RNA, rRNA. RNA polymerase 2 transcribes messenger RNA and some single nucleotide RNA. And RNA polymerase 3 transcribes tRNA and some other small RNAs. Each RNA polymerase has a specific promoter that it uses in order to transcribe. So initiation of transcription requires a bunch of transcription factors, which are proteins and enzymes that attach to the transcription site. And once they are there, then RNA polymerase 2 will bind and start initiating the gene expression. And when they are together, it's called the initiation complex, and it's at the promoter region. Termination sites aren't as well defined. There's still research going into that, and they are different depending on different types of transcription we have going on. Once your messenger RNA are made, you have a couple modifications made. One is called the 5' cap, and that is just a bunch of phosphate molecules that are put on the top, and it keeps the mRNA from degrading. It also has a poly-A tail, which is made of a lot of adenines, and it also protects it from degrading. And is important for knowing the difference between the head and the tail. You have a cap and you have a poly-A tail, so you know the front from the beginning and how it should be read. So once an mRNA is made, though, it's usually not all of it is used for transcription. You have introns, which are sections of the RNA that are used, and you have exons. Sorry, introns are not used, and exons, which will be used for translation. So what happens to the introns is they are removed by small ribonucleoprotein particles, or SNRPs, and they recognize the boundaries between the exons and introns and then cluster with other proteins to form a spliceosome and remove the introns. And once you have these exons identified, they can be mixed up. You can have 1, 2, 3, 4, that'll produce one type of protein, or you can do 1, 2, 4, 3, or you can do 1, 2, 3, or 1, 2, 4, and that's called alternative splicing. So it's the arrangement of these exons which also helps determine what type of protein it is. And this explains how 25,000 genes can produce more than 80,000 different types of RNAs. tRNAs then carry amino acids to the ribosome, and then the ribosome puts them all together to make a polypeptide. Aminoacyl tRNA synthetase adds amino acids to the acceptor stem of the tRNA. Okay, and then there's an anticodon loop, which contains 3 nucleotides, which are binds to the mRNA. So again, just like how A and G and C and T bind together in DNA and RNA, they have the same base pairs which will bind to the messenger RNA. Okay, and each one only recognizes one amino acid but has several different tRNAs. A charged tRNA is one that has energy from ATP, and then it can form the peptide bond using the energy from ATP. And the ribosomes don't verify the amino acid attachment to the tRNA, so if it's the wrong tRNA on there, the ribosomes won't be able to tell. Okay, there are a few binding sites. The P site is the part in the ribosome which attaches the growing peptide chain. So that's where the next amino acid is going to bind to. The A site carries the next amino acid, and the E site is where the tRNA exits. So there are 2 primary functions of the ribosome. First is to decode the mRNA, and second is it forms peptide bonds between them. And peptidyl transferase is the enzyme component of the ribosome which forms the peptide bonds between the amino acids. Okay, so translation then requires an initiator tRNA charged with informal methionine, which is a little bit different than a eukaryote. But this is then the first amino acid in all prokaryotes, prokaryotic proteins. Then you have a small ribosomal subunit which attaches to the mRNA strand. And then the ribosome binding sequence of mRNA positions the small subunit in the right place, and the large subunit attaches on top. So you start with the informal methionine charged transfer tRNA. It attaches to the small subunit, and then also the messenger RNA, and then the large subunit attaches on top. And now you have transcription ready to begin. Originally it is bound to the P site, and the A site is empty. It's very similar in eukaryotes, except the amino acid is methionine instead of informal methionine. And it's more complicated initiation complex rather than just a few simple functions. You have a bunch of transcription factors all acting, or translation of proteins all adding at the same time. And it doesn't have an RBS. It has a small subunit that binds to the 5' cap of the messenger RNA. Elongation then is the process of adding another amino acid. So one amino acid comes in there, it forms a peptide bond, and then it spits it out. The next one comes in. And this process just continues in a circular fashion until you have a very long chain of amino acids, and it comes to a stop codon. But there are fewer transfer RNAs than codons, and that's because that third position there allows for wobble pairing. Basically, that third part of the codon does not have to be a perfect fit. And that's why multiple anticodons can attach to a different codon, and it requires less tRNAs. Okay, so once it's all done, the elongation will continue until you come to a stop codon, and then termination occurs. They have release factors which release the polypeptide from the ribosome, and also and then there are no more tRNAs that are coming in and adding. So your polypeptide chain is complete, and then the ribosome breaks apart and will be able to go and translate another protein. So when things go bad, of course, we have what's called a mutation. There are point mutations where only a single base is being altered. Or you can have a substitution, which is a point mutation, where one base is substituted for another. It's a silent mutation if it doesn't affect the amino acid. So multiple codons, remember, will code for different amino acids, so it's silent if it doesn't change. A missense mutation is where the amino acid will be changed. And this will lead to either a transition or a transversion. A nonsense mutation is where it's changed to a stop codon, and that will cut the polypeptide short and will be non-functional as well, usually. A frame shift mutation is where you add or delete a single base, and that will throw everything off. So here you have a specific sequence of mRNA being converted into these amino acids. All you have to do is delete one thing. CAT, TCA is the same, CAC is the same. Oh wait, no. Oh, so if you delete that C and then you started at ATT, it would have a frame shift mutation, and all of these would code for different amino acids in these guys. So it doesn't take much of a deletion to throw everything off. There are also chromosomal mutations, so things that happen with the chromosomes, not with the individual sequence of DNA, and it changes the structure. You can have deletion, where we talked about chromosome 15 being deleted and how it could cause different disorders, whether it comes from a mother or father. That's basically where something happens and part of the chromosome is completely lost. You can have duplication, where part of it is copied, and you can have an inversion where it's flipped upside down, and that does affect things as well. Or you can have part of a chromosome splitting off and going and being inserted into another part of the chromosome. All of these will cause mutations, and you can identify, for example, the difference between a chimpanzee and a human is a inversion of one chromosome. And mutation is then the way that genes are altered in the way evolution occurs. It's mutations if they are advantageous. Most mutations are not advantageous. But if a mutation occurs in an advantage to a selection of nature, natural selection, then it will increase in frequency and cause a change in a species. So there must be a balance between these mutations that cause harm and are lethal and those that may be advantageous. And that's all for DNA and genes.